Welcome to Beyond Lab Walls, a podcast from the Salk Institute. Join hosts Isabella Davis and Nicole Milnarek on a journey behind the scenes of the renowned Research Institute in San Diego, California. We're taking you inside the lab to hear the latest discoveries in cutting-edge neuroscience, plant biology, cancer, aging, and more. Explore the fascinating world of science while listening to the stories of the brilliant minds behind it. Here at Salk, we're unlocking the secrets of life itself and sharing them beyond lab walls.
Hello listeners, I'm Isabella, your host today for Beyond Lab Walls. I got to sit down this month with Joe Herdy, a postdoctoral researcher in the lab of Rusty Gage, a professor and neuroscientist here at Salk. Joe took a somewhat slippery, maybe even slimy route to San Diego thanks to some lamprey research that helped him transition from plant science to neuroscience. Now, with lab mates in the Gage lab, he's uncovering the genetic drivers of Alzheimer's disease and other neurodegenerative disorders. Our conversation goes from exciting things like zombie cells into jumping genes and how they may relate to inflammation and the neurodegeneration that we see in Alzheimer's. So let's get right into our conversation. Joe, where did you grow up and what were you like as a kid? Were you always a little sciencey?
Yeah, I grew up in Huntsville, Alabama, which is kind of like a sciencey town, I guess. My family originally moved down there because my dad is an aerospace engineer, so he was involved in designing engines for rockets and jet propulsion and those sorts of things. And there's in Huntsville, Alabama, that's where they designed the Saturn V rocket, which is like the rocket that landed on the moon. So Huntsville has a lot of engineers and NASA nerds and that kind of demographic of people. And so it was like a very sciencey type town. And I wasn't necessarily as interested in the engineering part. I always sort of was more drawn to the biological sciences. I spent a lot of time in the woods. I was flipping over logs and catching snakes and salamanders and then bringing them home.
When did science go beyond an interest into a serious consideration for a career?
I would say that, like, pretty early on, I understood in more abstract terms. There were like doctors and lawyers and nurses, and there were also scientists. I would say as a kid, I didn't know like what it meant to go to graduate school or to have like a PhD or any of those sorts of things. But I did have an understanding that, yeah, like a scientist was something that you could be. And like my mom always tells a story that like the first job I ever said, or like for years, when someone asked me like, what do you want to be when you grow up? I would be like, oh, I want to be a paleontologist because I want to go. Kids love dinosaurs, obviously. I knew it's like, oh, this is something that someone could do professionally, and it's related to dinosaurs and science. Somehow it's like, that sounds like the best possible scenario is to be a paleontologist. And then after that, I remember I would like, in middle school, I was like, oh, I'm going to be a marine biologist. And I was living, another classic. I didn't really have like, like a real understanding of like what that entailed or what I would actually be doing. But I was like interested in like, oh, I'm going to go out and like catch fish and like understand like what it is that fish are up to out there. Yeah. And then I think it was when I started in like right before like in high school, before I started like my undergraduate studies at the University of Alabama, that was like around the time that the human genome was first published, or like when the Human Genome Project was like coming out. And that was what started me on the trajectory to where I am now. I thought that this was like really fascinating, that they had like figured out all of the genetic code for like people, and read some of these books like related to the human genome and just sort of discussing like human evolution and how our genes impact so much of like who we are as individuals. So at that point, sort of like at the end of high school going into undergrad, I was like, oh, I want to do like genetic stuff and like understand genetics.
Can you explain the difference between DNA, genetic code, and genome?
Yeah, sure. So there are similar terms. The genome is made up of DNA. So DNA is 4 letters, A, C, T, and G. And these 4 letters and thousands of different combinations is what leads to the information for making life on Earth, essentially. It's the common alphabet that bacteria all the way up to humans share. And then when we refer to the genome, that is what would we consider to be the genetic code, like specific to, I mean, it can be specific to an individual or a group of individuals, like a population. So we might say that someone has sequenced the seagull genome or the baleen whale genome. And that means that they have just figured out all the sequence of letters that is specific to that organism. But the interesting thing about genetics is that although in broad strokes, the genomes between individual people are much similar than the genomes between like a person and like a fly, there's also like a lot of differences between individual groups, like within organisms or like within different species. And so the genome can like be different on lots of different scales. And then genetic code, I guess that's when you're getting more down into like the nuts and bolts of like how the genome is like read and interpreted, because there's lots of kind of rules in terms of how DNA is read or interpreted by a cell, and how the DNA is like turned into proteins or like turned from like an individual cell into like a whole person. And so the genetic code, I think, is more related to like regulation on that and how your DNA actually ends up producing a different person.
That's super helpful. Yeah, so this is what really piqued your interest. And since then, you've ended up in neuroscience. What's the connection there?
My route through science was very, very meandering, for sure. Because at the University of Alabama, they didn't have any kind of like neuroscience or stem cell program. But there was a lab I was in, Leland Cheka's lab, and they were studying the genetics of invasive grasses. So like a lot of people know invasive species, they cause problems with the environment all over the world. And in Alabama, they had this problem with 2 different types of grasses. There was the wavy leaf basket grass and Kogan grass. And both of those are like, especially Kogan grass, like you see it at Home Depot all the time. It's a very popular landscaping plant. And it's sturdy and it looks nice. And so people buy it. They like to put it in front of their house in Alabama. But the problem with Kogan grass is that it can hybridize with some of the local grasses, and then it became like this sort of like invasive species. So it was taking over big swaths of like the forest in southern Alabama, contributing to like wildfire risk because it burns. It like the whole understory is like now filled with like Kogan grass. And basically that lab was involved in kind of like tracking the spread of these hybridized grasses. And so people would send us, we would get little plastic bags of grass blades. We'd grind them up, take the DNA out of them, and then we had these different types of probes we could use to look to see if it was like, did it have the DNA of like the native grass, or did it have the DNA of like these invasive grasses? And then so that was my work in undergrad, purely genetics, all plant biology, no neuroscience at all. And then after that, I did a master's degree at the University of Kentucky in Jeremiah Smith's lab. And in Kentucky, we studied the sea lamprey, which I don't know if you've ever seen a picture of a sea lamprey before. They're really like terrifying looking. They're like one of the most probably terrifying looking like sea animals that's out there.
Aren't they those eel looking guys with sharp teeth?
Yes. So they're the eels. They have these really sharp teeth. They don't have jaws. They're totally jawless. And they're not even technically eels. They're like 600 million years old. So like evolutionarily, they're super, super old. But Lampreys are invasive in the Great Lakes. They like parasitize trout. So they have like these really gnarly sucker mouths. They grab onto the side of fish and they like suck their blood, essentially. And we would get them, people in Michigan, like the state of Michigan is like really hates the lamprey. And so they would send any lab in the world. They'd be like, we'll send you this giant cooler where you'll get these big, like straight up like cooler you'd have like your beer in, but you would open it up and it'd just be a bag like full of like lamprey. And I don't even know how they would survive like the shipping, because they would like ship it to us, like USPS would come drop this cooler off. You'd open it up like, oh my God, there's all these fish in here. Because you can't breed them in a lab because they have to parasitize fish as part of their life cycle. So you can only get them sort of from the wild. Yeah. One of the many unusual things about sea lamprey is that talking about like genomes, how you have like the genome, it's like all of the DNA that is within an individual cell, like an individual's body. And usually when we're talking about genomes, we consider every single cell in our body has the exact same DNA, because at one point we were a single cell from like sperm and egg that fused, and that cell divides millions of times, and you have like a whole person. And one of the cool things about lamprey is that their DNA actually isn't the same across their entire body. They undergo this process that they described as programmed genome rearrangements. So this happens when the lamprey are developing from like an individual cell into a fully grown lamprey. So like when they're around like 1,000 cells, they delete 20% of all of their DNA from all of the cells that are gonna like become their body as an adult. And that little extra bit of DNA stays only in the cells that are going to become their sperm cells and their egg cells, so that it can get passed on to the next generation.
That's so weird. Which
is very weird, yeah. It's like a very unusual strategy for organizing your genome, because like in us, there's like parts of your DNA that you don't want to be expressed all the time, that you don't want to be turned on. And like famous example of that would be like cancer. When you have cancers, this means that oftentimes certain DNA that should be turned off is actually turned on. And then the lamprey strategy for dealing with this seems to be, well, we can't accidentally turn this DNA on if it's like not there at all. So they delete all of this. And the stuff that gets deleted, it's enriched for like these cancer genes and things like related to like cell proliferation, cells growing, and things like that. So the hypothesis is that, okay, this deletion is related to preventing cancer in some way. So, this is basic science. They're like, this is a cool discovery. We're trying to figure out like how this works and how they do it. And because of that work, my boss at the time, he invited someone from the Salk Institute to come and give a talk at the University of Kentucky, because they also studied this phenomenon. So Rusty Gage, my boss here at the Salk Institute, he studies like kind of a similar process. So when we're talking about differences between the DNA of individual cells within a person's body, the lamprey have like a really extreme version of this, where they have like 20% of all their DNA deleted from like all of their cells. But in humans, we discovered that, and he published a paper on this back in 2016, that there's actually differences in the DNA between individual neurons in our brain. So like the neurons are the electric cells in your brain that like kind of are firing electrical activity and control our whole entire like conscious experience and like all of our behavior. And one of the interesting things about neurons is that they have really very diverse morphologies, very diverse functions. There's like billions of them in your brain, and it's been a big mystery for a long time about how you could have such diversity within this organ, but everyone is starting with the same DNA. And one of the theories that Rusty put forward, or that he sort of discovered, was that actually there are differences between the DNA, between these neurons in your brain. And this is mediated through one of these jumping genes. So there's a class of genes called transposable elements. Some people call them like selfish genes, or they're like unique among all the genes in humans in that they can like copy and paste themselves. So over millions of years, they've copy and pasted themselves like a lot. So a lot of our genome is kind of made up of like these old fragments of like the copy and pasted gene. But they're still active, they're still with us, and that copy and paste mechanism happens in us as we develop. And that causes there to be differences in the DNA between neuron A and neuron B in your brain. And so, yeah, that like basically then I like met with this postdoc who came and gave a talk at the Salk Institute. And I was like, oh, this sounds really cool. Maybe I'll see if there's like an opening in this lab where I could come and work.
Wow, that is so strange. And is this happening just in neurons or other brain cells? Do we know yet?
Yeah, well, it in theory can happen to any cell in your body, really. It seems like it happens more often in neurons than in other cells in your brain. Yeah. There probably are important examples of it happening in other cell types too, but at least in terms of a matter of like scale, it seems like neurons are one of the cell types in your body that have the largest amount of this event.
So is this the kind of thing, the copy and pasting, is that happening throughout our entire lives, or is it more concentrated to development in the womb or even when we're younger and our brains aren't fully developed yet?
Yeah. Well, it can happen throughout your entire life, but you're much more likely to observe when it copy and pastes earlier in your life, because you can imagine that when you're 8 cells, if you have a copy and paste that happens in one of those 8 cells, then that's going to get propagated out, and millions of cells then will have that edit. Whereas if it happened when you're already an adult and your brain has already developed, once neurons are born, they never divide ever again. So if it happens in a neuron later in your life, it can happen for sure, but your chances of seeing that one are much lower just because it makes up a much lower fraction of like all of your cells. And we can't exactly go and take someone's entire brain.
Do you think starting in genetics has added a particular dimension to the way that you study neuroscience that's helpful in the lab?
Rusty's lab is in the laboratory of genetics at Salk, so he definitely approaches like neuroscience from like a genetics perspective. So I think that to the field writ large, though, I think that, yeah, bringing in this genetics component is like really important. For years and years, people have been wondering, how does vision work? How does taste work? We need to figure out which neurons are responsible for processing these types of information, and to like define and describe these things, they would rely on like the morphology of the cell. And now as the field has like advanced and genetic techniques in the brain have advanced, people are like, oh, well, we found that there's this one type of neuron that expresses this one very specific gene. And so we can look for this gene in this part of the brain. And it's like it perfectly describes like this group of cells that are important for processing fear or something. And there's more and more cases of this now where people are getting much more sophisticated in their understanding of like different individual types of neurons in the brain based on genetics. So it's a fun time to be a geneticist in the.
Yeah, is that the kind of question that you're asking in Rusty's lab?
So in Rusty's lab, we're definitely interested in aging and neurodegeneration. A lot of these questions about like this copy and paste mechanism, these are like really important for your development. Like it's going to change how your brain like grows and becomes mature, and therefore how you process information and a lot of those types of things. But then we're like more towards, that's like the beginning of someone's life, and we're a lot of the focus of the lab now is towards the end of someone's life. So why is it that some people experience this really profound like dementia as they get older, as their neurons start to die off and they lose all of their cognitive capacity? It's like, and develop Alzheimer's disease, which is like, the most common form of dementia. And it's a really big it's like a huge question in the aging field now as to why some people get Alzheimer's disease and some people don't. So that means like there's a lot of different types of diseases where there's like a really strong genetic component. So it's like, okay, well, if you inherit this gene, then you'll almost certainly develop certain types of Parkinson's disease, for example. And there are examples of that in Alzheimer's disease. There's these mutations in certain genes that they discovered back in the 80s. And they're like, these people get Alzheimer's when they're really young, like in their 40s. And, but like 95% of the people that get Alzheimer's disease, they don't have these mutations. They have like what they would call sporadic Alzheimer's. So there's nothing genetic about them that would seem to like really predispose them to Alzheimer's, but some people get it and then some people don't. And so we study a lot of that in the lab right now.
So thinking about aging and neurodegeneration, can you explain to me what senescence is or what senescent cells are?
Yeah, we do a lot of work now understanding cellular senescence. So the senescence is an interesting field. It started back like in the 50s. These two professors, Hayflick and Moorhead, back in the day, they thought that cells could divide for forever. Like people thought that if you took someone's skin cells and you put them in a dish and you gave them nutrients and water and heat, that they would just divide for forever. And the paper, it's kind of funny because the initial paper that describes senescence, it's not like, it's on a completely unrelated topic. Like they were studying tumor metastases. So like back then, people didn't really understand how tumors like moved around the body. And they thought, oh, maybe tumors are actually they're like a little incubator for viruses because they're all immune compromised. And so there's something about the tumor where they're like a factory for viruses. And the viruses are leaving the tumor and going to other parts of the body and infecting other cells and causing them to become cancerous. So they were just taking media from tumor lines and putting it onto human skin cells and then seeing how many times they divided. So they're dividing these human cells over and over and over again. And like the very last figure in the paper, they're like, oh, actually, by the way, guys, like we saw like after we made these cells divide like 50 times over the course of like several months, that they just stopped dividing. There was like this big drop off in the number of times the cells would divide. And they were like, we'll call this cellular senescence. So that's how the term originally came to the cell biology field, is to describe this like irreversible growth arrest. But in the years since that work, people basically discovered that there were lots of reasons why a cell could senesce. Not just this overproliferation, but basically like any type of like injury that a cell could sustain that is like so severe that it wouldn't want a daughter cell to inherit by dividing. So there's lots of changes in like metabolism from the cells or damage to their DNA or like different types of like cancerous transformations too. Like if a cell kind of detects that it's turning into a cancer cell, it can senesce in response to this. It's kind of shut itself down. And so it has like a lot of important like utilities in our bodies as we in the majority of our life. But for reasons that like aren't super clear, as we get older, we're accumulating more of these senescent cells. Our body is not as good at clearing them as it once was. Now they're just sitting there for like a long time. And the persistence of these senescent cells within different tissues in our body is associated with a lot of dysfunction. Because some people describe these as like zombie cells. They're sitting there, they're like not quite dead, but they're not quite alive. And they're sort of doing their job, but they're kind of bad at doing their job. And they're also trying to communicate to their environment like, hey, I've come off the rails, like something is wrong. I need to be like cleared. So they release a lot of these inflammatory factors into their environment, trying to alert the immune system and like the surrounding niche. It's like, hey, like you guys, I'm toast, like come get me. But when we get older, they're not getting removed anymore. So they're just like chronically releasing this stuff around, and this degrades their tissue that they're in and causes these issues. And so in our paper that we published back in 2022, because of sort of the history of senescence and how it had originally been described in like these dividing skin cells in a dish, people for a long time thought that it could only happen to dividing cells. And as I was mentioning earlier, neurons, they don't divide. But we sort of make the case for, actually, because there's all these other avenues outside of this like proliferation that can make the cell senesce, we think that these neurons, especially in the Alzheimer's disease brain, are entering into senescence, and that they're causing the inflammation in the brain that's leading to the neurodegeneration.
Okay, so you said you published a paper about that in 2022. What are the follow-up questions to that? What do you ask now?
I think the biggest one right now is really the inflammatory piece of it. I think we've pretty well established that there's senescent neurons. There's more of them in the Alzheimer's disease brain than there are in like healthy brains, and that they're releasing these inflammatory factors that are causing mischief. But neurons they don't really have like the machinery to produce a lot of these inflammatory factors. Like one of the most well-studied inflammatory factors are these things called like cytokines and chemokines, and neurons release those at their synapses, which are the connection between two individual cells, but they don't produce like large amounts of these inflammatory factors under normal healthy conditions. And so we were like, okay, well, we know these cells are becoming senescent, but how are they actually kicking on all of this inflammatory gene expression? And this is where we came back to the jumping genes, actually. One of our favorite theories is sort of the thing we're pursuing right now, is the influence of these retrotransposons or jumping genes on the development of inflammation. Ultimately, these genes, at some point millions of years ago in our history, were derived from a virus. It starts out as DNA in the genome. It's turned into RNA. It goes outside of the genome into the cytoplasm of the cell, like, the part of the cell that isn't contained within the nucleus. And then it undergoes a very unique process called reverse transcription, where it takes from an RNA, it can make a DNA, which is kind of like the reverse of how it usually works. Usually like DNA makes RNA, and then RNA is used to make protein, and the information doesn't go the other way. When the cell sees this RNA DNA hybrid, or a single lonely fragment of DNA out in the cytoplasm, the cell's like, oh my God, I have a virus infection. There's like a virus in here. I'm infected, and I need to respond to this. And so when they see this jumping gene that has this like viral-like intermediate, it's interpreted as an infection, and so it elicits an immune response. And this causes the inflammation. And we think that in senescent cells, they're having unusually high level of this jumping gene activity, which is causing them to do the inflammation. And this is actually, I think, a really exciting area of research because, like I was mentioning for HIV, they have these drugs that can block reverse transcription. Like people who have HIV, they take these drugs for decades to prevent reverse transcription from happening. So we know they're safe to give to humans. And we have some evidence now, or what we're working on now is like trying to target this process from these jumping genes to prevent them from making this intermediate to cause the inflammation.
That sounds like such a promising overlap there. It's making me think, of course, of Dimitri, who's an associate professor at Salk, and he does a lot of work with HIV. And it's cool to just think about those connections between genetics and neuroscience and immunology, but also the human connections between labs and people. I'm curious if you've noticed the neuroimmunology initiative at Salk having any impact or fueling your work.
Well, I've definitely noticed, I think like in the lab writ large, we've definitely noticed a lot more interest in like the neurome component. Yeah. And I'm not an immunologist. When I like have gone down like this path of like immunology, I'm like, oh my God, this is like sort of like a terrifying field. I'm very like naive. But right now, we're still not fully sure who is the one who's responding to these inflammatory signals. we have some evidence that like at least one other cell type in the brain, astrocytes, which aren't really an immune cell, they're like a supportive cell in the brain. We know for sure they can respond. We want to do experiments in these cells called microglia, which are kind of like a resident immune cell in the brain. We want to run some experiments to see how they are responding to these like neuron inflammation. But there is, I think, a huge like open field for other neuroimmunologists now, where people are discovering all these interesting types of immune cells that penetrate the brain. People know that like in Alzheimer's disease, you have like a leaky blood-brain barrier. And it seems like as you get older, and especially if you have like neurodegeneration, your little shield for your brain starts to break down and different immune cells get in that shouldn't normally be there. And so it's like, oh, well, what happens if someone not only has like these T cells or B cells getting in that shouldn't usually be there, but they also have this population of neurons that are suddenly releasing these inflammatory factors that they ordinarily shouldn't. Maybe it's actually the interaction between these two different things that is causing the degeneration. But as of right now, we're very much on the neuron side, like trying to figure out how it is that the neurons themselves are doing it, and then in the future would like to transition to like who's responding to it.
And is that related to, or could you tell me about your Bright Focus Foundation project?
Yes. So yeah, that's on my Bright Focus research. The Bright Focus is a organization that funds, like a philanthropic organization that funds different research initiatives into Alzheimer's disease. They have like a long pedigree of like important discoveries for Alzheimer's disease. And they have calls for postdoctoral fellowships. And so we produced some preliminary data, like kind of indicating that, we think these jumping genes might be involved in the development of this neuronal senescence phenotype, and applied to get some more money for research through this organization. And they thought that our ideas were good enough that they were willing to give us two years of funding to study this further. So it's been really exciting getting to work with them for this.
Yeah, that's a very, very cool. I'm excited to see what you discover. Outside of science, outside of the lab, do you have any hobbies that aren't science related?
Yeah, plenty of non-science related hobbies. San Diego is a great place for people. I was saying, like, as a kid, I liked spending time outdoors. Lots of opportunity for doing that here in San Diego. So a lot of times when I have time away from lab, like to go do the classic, go do some surfing. I'm not very good at surfing. Like, I'm actually pretty bad at surfing. If I can stand up on the surfboard, it's like a really great day for me. I'm like, yeah, I did it. I stood up and like actually caught a wave. But it's fun to be out there in the water and go rock climbing, go doing backpacking in the area, go hanging out in the desert. Down here, lots of opportunities for camping and things in Anza Borrego. And San Diego has a nice music scene. Like just this last Monday, we went to go, me and actually another postdoc in the lab, we both saw King Gizzard and the Lizard Wizard playing at the Shell. The Shell is a beautiful venue. Every time I'm down there, like this has got to be like one of the best venues in California. It's always nice there being like out on the water.
Was the ocean or surfing something you ever did growing up, or is that all brand new?
Yeah, surfing was completely new to me. Because growing up in Alabama, when we would go to the ocean, we would go down to like Pensacola, Florida, and like Navarre Beach and like that area. I love it down there. It's beautiful, like the whitest sand that's in the world, like these gleaming white beaches. And then the beach scene in the Gulf, that like panhandle part of Florida is definitely different than out here. Because I was like, when growing up, I'd be like, oh yeah, when I would go down to the beach, we would go like fishing and stuff like that, and then people like post up on the beach with their coolers full of beer and they're blasting country music, and they got their palapas like sitting there. And then out in California, I'm like, oh man, everyone's like running around and people are throwing footballs and people are out running in the waves and people are surfing on boogie boards, and oh my God, that guy's like, he has some sort of like sailing skateboard thing. Like people are so much more active on the beach.
So active, so much energy, really guilts you into getting up and doing something. My last question for you is, looking into the future, what do you think is the most exciting thing happening in the next few years in your field?
I think that, to me, the most exciting thing in the field is how a lot of some of the new technologies that are coming on board are going to let us answer. They're going to remove a lot of the barriers that we had in like asking some of these questions about transposable elements. So one of the hard things about studying transposable elements, these genes that copy and paste themselves, is that there's tons of copies of them in the genome, like millions of them. So when you want to look at the RNA to determine if these genes are being turned on, if they're being expressed, then the kind of traditional method would be with what they would call next generation sequencing. And they have what we would call like short read sequencing. So they basically take your RNA. So you can imagine like if you had like a phone book, you tear it up into like thousands and thousands of pieces, and you take each piece that is like 150 letters at a time. And then you can see where the overlaps of these different reads are. Okay, the end of this read aligns with the start of this read, which aligns with the end of this read, start of this read. And then you can kind of string together the actual full sequence of letters and like know which genes were being expressed. But this doesn't work super well for transposable elements or anything in the genome that's repetitive, because this short 150 read, you're like, oh, does this belong to this one that's here on chromosome 4, to this one here on chromosome 10? And so there's always all this like ambiguity for measuring the expression of these genes. But now the technology has advanced, and we have what they call long read sequencing. Now they can do entire messenger RNA sequencing. So now, like, instead of chopping all of your RNAs up into little bits, you're like, I'm just going to sequence the whole entire thing. And now this then removes the ambiguity from it. I know that it maps exactly back to this one particular copy in this one particular spot in the genome. And this just removes a huge barrier, I think, to a lot of people that like sort of exist in the field. And so it'll be really fun to see in the next couple of years as more and more people start doing this, like what everyone discovers with this new technology.
Yeah, I also feel like one of the more interesting applications for me personally of AI in science is in genetics, because there's just so much data and so many patterns. And I think that it's really cool to see new tech helping solve this old problem.
Yeah. I mean, the AI piece of it is pretty amazing, but it can be applied in all these like really powerful ways. And then in the genetics field, we kind of have a leg up in a lot of ways, because for 20 years now, people have produced way more data than you could ever analyze yourself. Like when we run one of these big DNA or RNA sequencing experiments, you'll get in like 130 gigabytes of data from all of your patients that you sequenced. But like, you just don't have like the time or the resources to be able to process everything all together. Whereas some of these AI tools, they're much better at handling like huge amounts of data like that, and can sort of like uncover sort of hidden structure that exists within data that we wouldn't, as mere mortals, like time to like fully understand ourselves.
Yeah, totally. We're in such a cool spot with lots of modern tech that I think is really going to accelerate a lot of different science. I'm really excited to see what you learn in the coming years, and I think it's going to be really surprising. But thank you so much, Joe, for sitting down and chatting with me today.
Yes, thanks for having me. This is great.
I don't know about you all, but I'm still stuck thinking about the fact that my neurons don't share identical DNA. These incredible discoveries are only possible when we have dedicated scientists like Joe on the case. I'm especially excited about his new Bright Focus project and the idea of getting inspiration from successful HIV medication to potentially treat inflammation in the brain, since both are related to retrovirus mechanisms. As Joe was saying at the end, modern tools and technology, not just AI, but things like long-read sequencing and new model systems, they're really invigorating the field. In the coming years, we can expect to learn unexpected, compelling, boundary-breaking information about the brain and about what goes wrong as we age to cause disease and disorder. And these are the stepping stones we'll use to reach treatment and prevention solutions for Alzheimer's, Parkinson's, and the many other neurological conditions we face today.
Beyond Lab Walls is a production of the Salk Office of Communications. To hear the latest science stories coming out of Salk, subscribe to our podcast and visit salk.edu to join our new exclusive media channel, Salk Streaming. There, you'll find interviews with our scientists, videos on our recent studies, and public lectures by our world-renowned professors. You can also explore our award-winning magazine, Inside Salk, and join our monthly newsletter to stay up to date on the world within these walls.